This is Dr. Robert Miller. Welcome to new advances into the very nature of the healing process and a discussion of clinical strategies to compress treatment time in oral implantology. Leukocyte-containing platelet rich fibrin, a second-generation autologous biologic used to enhance clinical outcomes in implant surgery. But what is L-prf? And how does it differ from other blood platelet concentrates? Simply put, PRF is a means of fractionating whole blood to harvest the bioactive components. It is a simplified chairside procedure, allowing us to fabricate thin compressed layers that are strong and suitable for suturing. It also promotes efficient cell migration, cell chemotaxis, and soft and hard tissue proliferation. It is a fibrin network rich in both platelets and leukocytes, and is reported in the literature to produce rapid healing not only during the first 7 days, but well beyond. PRF requires no anticoagulant, no bovine thrombin, no incubation, pipetting, or secondary spin. There are no chemical additives, and there are no expensive consumables. In fact, the cost per case averages less than $10. The fibrin in PRF is a protein polymer mesh. It traps regenerative cells during subsequent wound healing. But what are the benefits of L-prf? When platelets are released into circulation, they form something called activated thrombocytes. These thrombocytes produce growth factors such as transforming growth factor, platelet-derived growth factor, vascular endothelial growth factor, and important coagulation glycoproteins such as thrombospondin, vitronectin, and fibronectin. In addition to platelets and fibrin, PRF is rich in leukocytes. These white blood cells play a critical role in the healing process as mediators of the inflammatory and vascularization process. But why leukocytes? Leukocytes have anti-inflammatory and anti-infectious effects. They are also the most powerful promoter of vascular endothelial growth factor, which is responsible for the early generation of a rudimentary blood supply. There are applications in dental and oral maxillofacial surgery, including bone defects, extraction sockets, sinus and dental ridge augmentation, palatal defects, and maxillary bone atrophy. PRF acts as a carrier for particular bone material. When incorporated, the graft material is suspended in the fibrin matrix, and handling characteristics are dramatically improved. PRF is fabricated chairside with the Intraspin system by Intralock International, the only medical device that is FDA cleared for the production of PRF. It is a simple 3-step procedure. We draw blood, we spin the blood, and then we express the fibrin clots. In this video, we see a single fibrin membrane holding multiple heavy surgical instruments. This really speaks to the durability and strength of the PRF membrane. In fact, it can be sutured and it will not tear. There are times when we may prefer to have socket plugs rather than membranes. Here we demonstrate the fabrication of a socket plug. We will place the fibrin clot into the Delrin cylinder, and then we will compress these clots with the piston assembly. The piston assembly is inverted and placed into the Delrin cylinder and compressed until it is even with the top. In this way, we fabricate very consistent plugs that are suitable for extraction sites. Here, the fabricated fibrin plugs are demonstrated. There may be times, however, that we need to employ a biomaterial. We can use this serum that is collected at the bottom of the collection tray to hydrate the bone graft material. In this way, we make the bone graft more bioactive and we improve the handling characteristics. Note the adhesion of the bone graft material to the spatula. PRF is also an excellent carrier for particulate bone material. It can be used in one of three ways. We can mince it and mix it with a bone graft material. We can place a bolus of graft material surrounded by a PRF membrane, or we can mix the particulate within the PRF matrix before it coagulates to make a bone patty. Leukocyte containing platelet rich fibrin. Growth factor technology available from the scientific research team dedicated to biomimetic implant technology, Intralock International.